and please take it away. Thank you. Thank you much. So I'm going to tell you how we've been using the KCs at OEHA so far, and here's my conflict of interest statement. I have no conflicts of interest, and I do have a disclaimer here, very similar to Javier's, but mentioning my agency and department. So there's two main ways we've used the KCs so far. And here the slide talks about the first, which is we looked at the ToxCast and Tox21 assays. And even before the KCs came out, we wanted to map some of those assays to cancer pathway related, what we knew about cancer pathways. And so Shobha Iyer was the lead on this manuscript. She spent a lot of time, just takes a lot of time to do the mapping, even for cancer pathways. She looked at 3 different assay platforms within ToxCast. And then after mapping the cancer pathway related assays, she took that subset and mapped them to the KCs of carcinogens. And so that's one way we've used it. We've done a similar thing. The second bullet there, it's Jennifer Shea's work, primarily. Again, looking at omics data and trying to map using a whole variety of tools, the different changes in gene expression to cancer pathways, and then mapping those to the KCs. So we've done some of that work. We've used that in our reports, but I'm pointing you to the publications. And the take-home messages from these 2 projects were that, well, one, we think you can do that, as has been said by other speakers today, you can do the same mapping approaches to the other sets of KCs for hepatotoxicants and immunotoxicants, et cetera. It may be very useful in male reproductive toxicants. Other messages are that the KCs, we think, can be very useful for hazard prediction, for chemical prioritization, and for assessing the extent of biological coverage of the NAMS assays. All these points have kind of been mentioned by others before me. So I'll move on to the other way that we've used this. We're using the KCs of carcinogens and hazard identification, both under the Proposition 65 program and the Public Health Goals for Drinking Water program. And we're also using the KCs of male reproductive toxicants and endocrine disrupting chemicals in our most recent document, hazard identification document for Proposition 65. So here, focusing on cancer hazard, this shows you the different streams of evidence in green for a particular chemical, for evidence of carcinogenicity, and then that last green bubble, mechanistic and other relevant data, we've expanded it out, and all the yellow bubbles are showing you the 10 KCs of carcinogens, and then we have other aspects of mechanistic and other relevant data. So we're using this scheme to organize our thoughts and the evidence to discuss in our documents. In the early days of using this, so from 2017 to 2018, we looked at 3 different chemicals, coumarin, gentian violet, and methylenamine, sorry, NHEX for short. And we did our usual search, not focused on KCs, but on looking for mechanistic data when we did these documents. But we had omics data we found, and we mapped those omics data to cancer pathway related genes, and then mapped that mapping to the KCs of carcinogens. We did the same thing for ToxCast and Tox21 data. And in discussing all the data, we tried to highlight in those documents data that was relevant to specific key characteristics of carcinogens. So we were getting used to using the concepts. And over time, we've gotten more used to it, and improved our methodology. So the most recent example is from September 2021, and that was our document on PFAS, shown here, the cover of it. And for that document, we used a systematic approach to search for studies in the literature on cases of carcinogens and other mechanistic concepts, with search terms that were based primarily on the NTP's report, on carcinogens handbook guidance. IARC also has a set of search terms, and we looked at both. And for that particular chemical, this set seemed to work. We may mix and match in the future. So once we had our search, and I'm going to say we're focused, we do different searches for different streams of evidence. So here I'm just talking about the search for the mechanistic data stream of evidence. We use the KCs to help organize, identify, present, and discuss the mechanistic data. And we found it very, very helpful for PFAS. And here's this wonderful schematic. We presented this in some posters at SOT this year, where we've tried to list, we've got all the 10 KCs in that circle there, and the ones with the dotted lines means we didn't have any data at all that informed one way or the other those KCs. The KCs that are bolded had more data that suggested, that was supportive of that PFAS had that characteristic, but we've kind of listed here what data we did find related to each KC. So we find it very useful to organize and present this data. In this particular instance with this chemical, we didn't have enough confidence to put together a real mechanism after looking at all the data, but you can see that PFAS does quite a few different things. We just don't know enough right now to put something forward that really makes a lot of sense. We've also used the cases of carcinogens in a public health goal document shown here. That document covered both PFOA and PFAS. It was released a little bit earlier than the document I just showed you, a couple months earlier. And we, since we had 2 chemicals, and we only did a select 5 of the KCs. KC 2, 5, 7, 8, and 10. If you don't know those by heart, it's genotoxicity, oxidative stress, immunosuppression, receptor-mediated effects, and cell proliferation and apoptosis. Okay, and so we did focused literature searches for data related to those 5 KCs for both PFOA and PFAS, and then summarize that in the document by KC. And now we're starting to use, for the very first time, KCs to evaluate chemicals for reproductive or developmental, reproductive effects. In the case, the example we had, we were looking at PFNA and PFDA, and we used the KCs, or we used them by highlighting the mechanistic evidence we'd found for those documents, for those chemicals, sorry, that were relevant to the KCs of either male reproductive toxicants or endocrine disruptors. So we're getting our feet wet with these KCs and trying to figure out what the best way is to present the data, organize the data, discuss it. So thank you.
Really neat to see this gray literature where